This conference will now be recorded.
Okay, recording has now started. So thank you all for calling in today. And please let any of your colleagues know if they were unable to attend that the recordings and the slides will be shared on our Southeast Region GitHub page, which I have the link at the end of the slides to share with you. So this training is going to be a 5-part training to go over SARS-CoV-2 data submissions. This has been a high priority subject for everybody. And okay, I'm sorry. So this is is everybody muted?
Yeah.
And then mute again. Okay, there we go. Okay. So each training will be a 1-hour training, and we're going to do parts 1 through 3 this week. By the end of parts 1 through 3, you'll be able to submit your consensus genomes to the appropriate repositories. We'll have some follow-up calls and then go through to the next parts. My slides are advancing. There we go. Okay, so as I just mentioned, 5 parts. Today is the general overview. Tomorrow we'll go over sample review, how to batch those samples for submission, and preparing your multi-FASTA file. Part 3 on Friday, we'll be going through, once you have all your files ready to go, walking through those submissions for GISAID and NCBI. After that, we'll do follow-up calls with individual states, if requested, to do hands-on walkthrough with you with your own data to make sure that you've grasped everything and you're having success submitting your consensus assemblies and sample metadata. After that, we'll go through the last 2 parts of the training where we'll do FASTA submissions to the Sequence Grid Archive, going through how to dehost those samples, pulling out any human reads that may exist. And then in part 5, we'll go over something that we do here in Florida. So if you get an error from a submission and they say that there's a flagged sample, I'll show you how to go through those flagged samples, how to address those if you wish, confirm new variants, and if you do detect any kind of PCR artifacts or sequencing artifacts, how you can correct those assemblies. So today we'll just give a general overview. So for today's outline, I'm going to first just give, we haven't had a call in a while, everybody from the region that can call in, so we'll just give some region updates and some HiPerGator updates. We'll go over just a general SARS-CoV-2 data submissions workflow overview, go over GISAID. I'm going to show you some examples of how to pull data from GISAID for your own use, and go through an NCBI overview as well. So a couple updates for the AMD Southeast region. So for this year, Florida and Georgia are the training and workforce development lead. Florida is still the BRR. We estimate that we'll have our annual week-long training in the spring, but obviously that's to be determined based on COVID. As you know, last year we were unable to have that due to COVID and not being able to travel. So fingers crossed that all will be well. But in the meantime, we'll continue having our online trainings and webinars like we do, and these can be one-on-one or group trainings that I've been having with most of the states in the region, and this can include things such as our Linux 101 trainings for new users, HiPerGator training, base-based command line interface training going over various scripts and pipelines for doing bacterial whole genome analysis. We have some scripts and pipelines for viral NGS analysis, mainly focused on SARS-CoV-2, but they're applicable to other viruses, so on and so forth. We do have a new bioinformatics software in Florida to help support my duties as BRR. Dr. Jashi Lee joined us here a few weeks ago, and he's a great resource that can help answer questions, provide code, whatnot, for the region. And we also sent out an email just a couple of days ago regarding increases to compute resources on HiPerGator for everybody. So I'll go over those into more detail. So effective today, each jurisdiction within our region now has access to increased compute resources on HiPerGator. We were able to increase what we had. So we now have access to 150 NCUs. So one NCU stands for one CPU and 8 gigs of RAM. So this means at any given time, we can be running up to the usage of 150 CPUs over 1,000 gigs of RAM to run our different jobs and analysis, which allows a lot of us to be running jobs at the same time, running multiple samples at the same time. For blue storage, so just a reminder, blue storage is like your main regular storage on HiPerGator. That's meant for high performance to be used as input and output for your different jobs that you're running. Each state now has 2 terabytes. Some states, based on your request or usage, now have more, and the states that we've been in communication with, you have more than the 2 of what was discussed, but other states, if you do need additional storage on blue, let us know and we can increase that. But right now, everybody has a minimum of 2 terabytes. And then we also added, this year we added orange storage. So orange storage is considered long-term storage. And by long-term, it's just data that you put into HiPerGator that you're not currently using for a current analysis, but that you need to keep on HiPerGator. Maybe you're part of a project or some of your SARS-CoV-2 sequencing data that you can put on the orange drive that's not currently being used for a run. So you have 10 terabytes. So in total, you have a minimum of 12 terabytes, not more, on HiPerGator. And I can go over later if you have questions on how to use the orange storage differently than the blue storage. Also, a reminder that HiPerGator is free, so it's continuing to be free for all Southeast region jurisdictions. With these compute resources, unlimited analysis. So it's not, there's been a lot of discussion lately, some regions use cloud computing, where you have to pay per sample, pay per analysis, pay based on the resources that you're using at the time. This is all inclusive. So you can rerun samples analysis as many times as you want. The only limit is that if enough people have queued up enough jobs to take up 150 CPUs at a time, it just gets put in the queue, and then as soon as resources are freed up, the next one gets run. And we've actually never hit a maximum before all at once, but that's the only limitation. So we have quite a bit of resources for everyone to be able to run the analysis if they need to, as much as they'd like. So also just a reminder that you can find all HiPerGator resources and information on our Southeast Region GitHub site, which I've included the URL here. We have the documentation up there. If you have anyone in your lab or you're new to HiPerGator and you need to request access and don't have an account yet, the information sheet is on there as well. You can also email me, ask me questions, I can send it to you. I have links to help and documentation and other training videos for HiPerGator that you can go to as well. And then all other past trainings and webinars that I've given in the past, including the recordings and the slides, are all in the webinars folder at our GitHub link as well, so you can access those there. And I'm bringing up all this HiPerGator information again because we're going to be using HiPerGator in order for a couple of the steps for the submissions to format our FASTA files and do our sample review. So this does require a HiPerGator account for the individuals doing the submissions. You do either need to already have command line knowledge or request the Linux 101 training from me. And I do that, it's just a 1-hour training. A HiPerGator training, so if you're new to HiPerGator and you haven't set up your account yet, we'll walk through together on a call, walk through our analysis reference guide we put together to help you set up your compute environment, make sure you have everything installed and that you're ready to go to run our different scripts and pipelines. And then also, have knowledge of our base-based command line training, which is a 1-hour training, just so that you know how to pull in your data from base if that's where you're pulling it from. You can pull it directly from base directly. So I'm just going to review again our current scripts and pipelines that are available for all state public health labs currently on HiPerGator in our region. So we have our just general FOC pipeline, which is a general de novo assembly imitation quality metric pipeline for whole genome sequencing of bacterial samples, generates a report in your annotated assembly. We have another version of that called FLOC AMR, which I know several jurisdictions in our region are using on HiPerGator to analyze any of your HAI organisms. So this does everything that the FLOC pipeline does. It also adds in species ID, runs MLST if there's a pub MLST scheme, identifies AMR genes, valence genes, plasmids, and if it's salmonella or E. coli, performs serotyping. It'll output all of the metrics that you need that's based on the ARL and WGS guidance that you have to report back or make sure you have a proper QC of your samples before submitting this to CDC, and that outputs everything for that. We also have included with the FAT pipeline, we have two of CDC's Legionella tools on HiPerGator. So this is their Legionella species ID tool and also their Legionella serotyping tool. We compare those to our lab serotyping results. Those are both on HiPerGator. We also have additional scripts for organisms where an unknown species classification and trying to get a species ID for something that is uncommon and failed to get an ID from basic culture. And so we have a few different tools and different databases all ready to go on HiPerGator for those needs. We also have a couple of SNP pipelines that we use. One is a reference-free method. One is a reference-based method. We're looking at whole genome SNPs and high-quality SNPs. So this will do multiple sequence alignment, output pairwise SNP matrices, and build phylogenetic trees. And then we also have custom scripts to annotate those trees for you. So those are helpful when we get analysis requests for EPIs, looking at trying to identify different clusters, see if something is included. We'll run those pipelines. We now have some viral pipelines and scripts. So we have a SARS-CoV-2 pipeline on HiPerGator that you can use. It's called FLAC SC2. It is compatible with Illumina-based sequencing using any of the Arctic version primers. So you can use it for Arctic version 1, 2, 3, or 4. It'll also work with, we tested out some tailed primers where it was like a non-Illumina library prep, and it works with that as well. And it also is now compatible with, if you've done your prep and amplification with the QIAseq assay, it's compatible with that. It's also compatible with COVID-seq assay because they use the Arctic primers. You just have to make sure with that you're using reads at least 150 base pairs long. And that generates a assembly report, your consensus assembly. It uses the latest version of Penguin to do a lineage call, and it also automatically runs GenBank's Vader viral annotation pipeline, and it gives you a QC flag, whether it passed or failed, and it also gives you a flag for Vader, whether it passed or whether that's a sample that you need to manually review prior to submission. We also have another script that works with, if any of y'all are generating SARS-CoV-2 data on the CLIA Labs platform. They provide an analysis method in the cloud, and then they give you back your assembly, your FASTQs, your BAM files, and your VCF files. We have a script that will then take in all those files to generate an assembly quality metrics report that looks very similar to our other pipelines. So you can compare it to your other data. It will do the Penguin lineage calls for you, runs Nextclade, and it'll automatically run Vader. So again, helping for doing that sample review process automatically as part of that pipeline prior to submission. And then during this training, we'll go over, we have a couple scripts that help automate the formatting of your FASTA files and doing sample review for SARS-CoV-2 data submissions, which we'll be going over in that training. We also have a script, we'll be going over this in part 5, that will provide assembly correction. So if you identify something that appears to be a PCR sequencing artifact that needs to be removed from the assembly prior to submission, we have a script that automates that as well. And then we also have some general scripts that just help with basic targeted amplicon variant and consensus generation that can be used for other viral pathogens. We've used them for HIV, so those can be used as well. And then we also have a number of individual scripts for running any kind of individual bioinformatics tool. You can use the StaPH-B toolkit on HiPerGator, run Nextflow workflows. We also have scripts that we use if we're generating any data on the NextSeq that generates FASTQs across 4 different lanes in your flow cell. That's incompatible with bioinformatics for pulsenet organisms. So we have a script that will concatenate all those to make them compatible for bioinformatics. So if you're doing any kind of pulsenet sequencing on the NextSeq, we can help you out with that as well. And then we also will write custom scripts and pipelines as requested or needed, which we've done in the past. If you also need help with any kind of analysis or support, you can email us. You can also send me an email to my individual email. and we can set up one-on-one video calls and go through that as well. Okay, so now that we've gone over that, so now we'll get into the specifics in the general overview of SARS-CoV-2 data submissions. So I just want to point this out really quick that APHL, as well as there's some other sites, have a collection of information related to SARS-CoV-2 sequencing, including wet lab protocols, information over genome submissions, bioinformatics tools, training resources in the CDC's S3 program. And I wanted to highlight for the genome submissions, they have a sequencing recommendations document where they provide recommendations for things like genome coverage and depth in regards to which samples you're going to submit and different criteria for the different repositories. This is currently the latest version that's online right now is from March. They're currently updating this version, so there'll be updated guidance and recommendations forthcoming, but I did want to point that out so that is a resource that you can go to as well. So I want to walk through just the general SARS-CoV-2 sequencing workflow and what kind of data you're getting out from that and what data is recommended for submission. So most labs are doing a tiled amplicon or enrichment-based approach. Many labs are doing Arctic-based sequencing using the Arctic primers, but there are some other kits out there, on the Illumina platforms, and some people are doing Minion nanopore sequencing. So you get your fast queues from that. Goes to your data storage. Illumina is probably going into base space. Others might be using some other kind of cloud repository, pushing that through an analysis pipeline. So here in Florida, and I know there's some other jurisdictions in our region using the Flock SC2 pipeline and HiPerGator. So all of your data would be getting pushed to HiPerGator. There's also the Secret pipeline developed by Aaron Young in Utah. There's the Illumina Dragon pipeline. So I know that a number of different labs are using different pipelines. You're going to get slightly different file types out from those pipelines in different formats. But all of that essentially is coming down to generating a consensus assembly, getting a lineage plot, and a final sequencing report. And there's usually going to be some type of variant or VTF file and a BAM file coming out of whichever pipeline that you are using. So from there, we'll then talk about what is required or recommended to submit. So from the output from any of those pipelines, it's recommended the bare minimum to submit your consensus assembly, both to NCBI GenBank as well as to GISAID, along with that sample metadata. So that sample metadata is going to go along with your submission to GISAID, but that sample metadata on the NCBI side is going to go into GenBank as well as into their biosample database. It's also recommended to submit your raw FASTQ files and BAMs to the NCBI sequence read archive. For these parts 1 through 3 of this training, we're going to focus primarily on the high priority submissions of consensus assembly and your sample metadata along with that. And then in part 4 of our training, we'll go through the FASTQ submissions to the sequence read archive. So I put together this kind of process or checklist of everything that's involved with submitting your consensus assemblies to GISAID and NCBI and making sure that your data is all linked. So essentially the submission process starts with, you're going to screen all of your samples once they come into your pipeline. You're going to screen for your passing QC samples for submission, and you'll need to run them through Vader, which is NCBI GenBank's viral annotation pipeline. And we're going to go through that tomorrow's training. We're going to go through that in more detail about what Vader is, what is it looking for, and whatnot. So you'll need to screen them to see which ones are likely going to get flagged or not, because the different databases have different criteria based on which samples will be accepted and which ones won't. And they're doing it based off of annotation pipelines, and some of them will get flagged and will get rejected, and it has in the past created bottlenecks in the submission process. So we always recommend pre-screening your samples first, which you can do on HiPerGator. We have a script for that, and batching your samples based on which ones pass QC and which ones pass the data review. You're going to select those samples for submission, collect your relevant sample metadata needed for that submission, needed to fill out all the different metadata templates. So in this case, there'll be 3. There's going to be one for GISAID, there's going to be one for biosample, and there'll be one for GenBank. And they all overlap with most of that information. You'll then need to assign the public repository sample names. We'll go over that in more detail tomorrow about what is required and how the samples need to be named. You'll need to prepare a formatted multi-FASTA file that has all of your FASTA sequences from all of your assemblies into one file that you'll submit to GISAID and a different one to GenBank. And we have a script for that to do that on HiPerGator, and we'll go over that in tomorrow's training. You'll then submit your FASTA file and your metadata template to GISAID. You'll wait for them to accept your samples. You'll then retrieve those accessions, which I'll show you how to do that on Friday. And then after that, you'll submit to NCBI biosample. You'll submit the metadata template, just an Excel sheet, with those linked to GISAID accessions. So that's why I recommend different labs will do this differently in different orders. But for us, I think this is the simplest way, is to screen everything first, submit to GISAID, get your accessions. That way you can include your accessions on the biosample sheet to make sure that they're linked. Submit to biosample, save your biosample accessions when they come back. They usually come back within like 10 minutes. And then submit to NCBI GenBank, where in the metadata template from GenBank, you can include your GISAID accessions and your biosample accessions, along with your FASTA file, and then save your accessions and you're done. That way, at the end, all of your data is linked, so that if someone's trying to look at your data, or you are, you can see from, especially on the NCBI GenBank side, you can link that to your biosample and to the GISAID. And I'm going to go over biosamples and whatnot in a little bit more detail in a second. So first, I've been mentioning GISAID several times, and just in case someone is not familiar with GISAID, I imagine that you are, but just in case you're not, I'm going to go over it really quick. So most people are familiar with NCBI, but GISAID, it stands for Global Initiative on Sharing Avian or All Influenza Data, and it started out of necessity to help track influenza data. It's now being used to track SARS-CoV-2 data, and I believe they've now started a database for RSV as well. I've included the link here, and I'll be doing a short demo to go over it to show you how to pull some information from that. So GISAID is a public-private partnership, nonprofit. It's in partnership with the governments of Germany, Singapore, US, along with private and corporate philanthropy. They have the EpiFlu database, which is to track influenza data. They have the EpiCog database, which is to track SARS-CoV-2. And I believe they have an EpiRSV database now. This database is a little bit different. They try to encourage the sharing of data prior to publication, and by doing that, ensuring that the submitters of the data maintain the protection of their intellectual rights of that data. So there's strict terms of use and data usage policies when it comes to GISAID. That's a little bit different than NCBI. If you ever use data from GISAID, you have to include acknowledgement tables in your publications. You are also you're also recommended and encouraged to reach out to the original submitting authors of that data to include them on any publications or any studies. And if you use their data, make sure that the focus of your papers and whatnot are included, or focus on your own data, but using others kind of more as contextual data. It's all in their terms of use. Currently, there's over 3.5 million SARS-CoV-2 genomes in GISAID. It is the largest repository of SARS-CoV-2 data in the world. It has more than NCBI. And I'll go over in a little bit why it's beneficial to submit to both and what the differences are. So for most of y'all are familiar with NCBI. I'm not going to go into all the details and all the different databases and all the different information and tools that are in NCBI. There's too many to go over, but I'm going to focus on the ones that are relevant to what we're doing regarding SARS-CoV-2 data submissions and data usage. So the three different NCBI databases that we'll be submitting data to are Biosample, which is a database that strictly just collects your specimen metadata. It has no genetic data in that specific database, but it links your specimen metadata back to genetic data in other databases. GenBank, database for nucleotide sequences. For example, this is where we're submitting our consensus assemblies. There's currently over 1.3 million SARS-CoV-2 sequences in GenBank. Sequence Archive is a database for raw sequencing data where you can put your FASTQs, you can put your BAMs. If you have nanopore data, you can put the FAST5 files in there. There's currently over 1.1 million SARS-CoV-2 data sets in Sequence Archive. I also included links here. I'm not going to go over them in detail, but to other NCBI SARS-CoV-2 resources. So the first link will take you to just their general SARS-CoV-2 resources on NCBI, where you can get SARS-CoV-2 data. Resources found at NCBI virus. which allows you to query different information, do different analysis on NCBI, use different tools. It's very helpful. And then there's a beta version of NCBI dataset, which is a new tool or database way of collecting different types of data from different NCBI databases to use for your purposes. Okay. So I've mentioned GISAID, NCBI, we need to submit to both. Why do we have to submit to both? So as I mentioned, GISAID is a public-private partnership versus NCBI is public and open data. GISAID has strict data usage policies in terms of use. They have things like you cannot download their data and then host that data elsewhere without their strict permission. They're also very strict about using their logo or anything that you pulled from GISAID. So there's just different rules in terms of use that apply. Versus NCBI is a public database. You can open that or you can download that data and use for whatever purpose. There's different features and different ways to query the data. Some are easier than others with the different databases. So you might find one more user-friendly than the other one. There's differences in acceptance criteria. So for example, for submitting your consensus assemblies to GISAID and to GenBank, they use different screening approaches. They use different bioinformatician pipelines. GenBank's Vader pipeline is open source. That's why we can run it ourselves. GISAID's pipeline is not open source, so we can't run it ourselves. They have differences in what they will accept and what they won't. We try to make it consistent across both databases of what you submit to GenBank, what you submit to GISAID is the same FASTA sequence and vice versa. If you ever run into a situation where one database might accept one sample and the other one doesn't, and let's say you need to go back and make a correction, you need to make sure to go back to the other database, email them, and send them the updated file, updated metadata, whatever it is that you had to change. Data are pulled from GISAID and NCBI for different purposes. So for example, for a lot of our next-gen builds, GISAID was allowing the next-gen team to provide a curated metadata file that was very already formatted to use as input for the next-gen pipeline. And they've now discontinued that. So now we're pulling data from NCBI GenBank to make our next-gen builds. So there's just different purposes for why you would pull data from each and how we're allowed to use that. So that's why it is highly recommended to submit to both and link your data across both databases. So prior to doing any submissions, you need to make sure that you have accounts for each database. So for GISAID, I've included the link here. So if you don't have an account yet, go to this link, follow their setup instructions, and you'll be able to get an account and make sure that you're able to submit data. NCBI, your lab should have an NCBI account for, you've been submitting that's a lot now for PulseNet, you're all in. If you don't, go ahead and make one. But prior to doing any SARS-CoV-2 data submissions, you will need to submit or register a bio project for your lab to make sure that all of your data in NCBI is linked and collected under this one bio project. That's also linked to the CDC umbrella bio project, and I'm about to go into that in more detail. So here's the link here. In case you have not registered your bio project, do that first before you do any SARS-CoV-2 submissions. And if you haven't yet, you should. You have to link it to different information. Go ahead and do it. Okay, we're going to go through those. So NCBI bioproject. Bioproject is another database on NCBI that can be used to kind of think of it like collect and organize all of your data associated with a specific project or a specific paper, specific purpose. And so in this case, it's going to link all of our GenBank, SRA, and biosample submissions all together. CDC has an umbrella bioproject for coded sequencing. I've included it here in bold. So when you go to the bioproject URL, it looks like this. In the red box down below, you can see the submission. Click on submission, that's where you create your new bioproject. Towards the end of the submission wizard, it'll ask you if this is associated with another bioproject. You include yes, and you include this CDC umbrella bioproject number to make sure that it's linked. And I'm going to show you what that looks like right now. Let me minimize this really quick. So We go to NCBI Bio Project. So if we search and copy paste this, this is the CDC umbrella bio project. You click it here. It'll take us to this page. So you can see all the data that is linked underneath the CDC's bio project. So they just have a description here, the accession, the fact that it's an umbrella project, which means that there are multiple bio projects linked to this one. And here's all the data that is associated with the CDC umbrella bio project. All of the individual sub-projects, so this will be like your individual state lab bio project, are listed under here. So I'll just show you Florida's, for example. We'll see like our title, SARS-CoV-2 whole genome sequencing, FLDOH. So like this is ours, we can click on it and you can see the data that's been linked into here. So we have 4,400 GenBank that are linked to this bio project. We have more than that, but I have to go back and link some of our older ones. We have over 9,600 biosample submissions, protein sequences. So all of your data will be linked underneath your bio project. And if you ever had an interest in looking at data from some of the other states, you can see that here's Georgia's, here's Wadsworth, here's University of Texas, Utah, Arizona, all linked underneath the CDC bio project. So that's why it's important to register your bio project so that you can link all of your data, but then also include it as the part of the CDC umbrella project. Okay, let me go back. All right. Okay. So for GISAID, I'm now going to show you a demo of GISAID when you log in, what it looks like. I like using GISAID. I think it's pretty user-friendly for being able to query data, for example, like from your state. We get a lot of questions a lot from our EPIs or from lab leadership or whatnot, asking like, how many deltas do we have in our state right now? You know, how many alphas or whatnot? And do any of those have particular mutations? GISAID provides a very user-friendly way to be able to pull that information. You can also get the same information from NCBI, but I've just been more familiar with GISAID from the get-go with that. You can also download data for downstream analysis from GISAID, both the FASTA files as well as the metadata and usage reporting. So we'll go into that. Okay, so GISAID website, gisaid.org. So the login button is here. There's a bunch of other information you can read about. And so once you have your account, you'll sign in. So this is what the page will look like when you first sign in. It automatically takes us to the EPICOG database. And you have a number of different selections here. So when we'll go over this on Friday, but when you're going to upload or submit data, you use this upload button. We'll go over that Friday. We'll go over this in a second, how you can download download packages, compressed files from here. But just to do simple search queries for data, you'll click on the search button. And this gives you quick stats to down here at the bottom of what is in GISAID. So right now you can see at the bottom where it says total, and this is 3,551,680 viruses. So there's currently over 3.5 million SARS-CoV-2 genomes in GISAID. That includes everything that's ever been submitted. So up here, you can use these different search features to search based on the information that you're looking for. They just have to be formatted in a particular way. So for example, if you're trying to get all the data from your particular state, go under location. They have it organized first by continent. You have to do North America, and you have to do, this is important, space, / another space, USA, space, / another space. And it'll start, it's a little bit delayed, but it'll start, as you can see, it starts popping up your different options. And I'm just going to do Florida as an example. We'll wait for it to go. Just a second. Yeah. Oh, it's accident, of course. Hang on, I'll wait for it to finish and I'll correct it. Some people have, we don't submit county level information, but some people or like academic groups. So, because this will have submissions from not only your lab, but anyone, so in this case, anyone from the state of Florida. So this will include our data. It'll include data from all the CDC contact labs that's been submitted by CDC, any academic groups in your state. It's not the fastest thing in the world. There we go. Okay, so for Florida, you can see at the bottom, so this gives you like a quick number of how many sequences we have from Florida that have been submitted by different users. So we have over 67,000 sequences from the state of Florida. So you can also start querying if you have questions over particular lineages. So you can see how many E1 617.2s we have, and you type that in. As you can see, it also, I find it faster to type it in than waiting for the arrow to come up. So we have over 18,000 E1 617.2s. You can also do other search features like this is giving us the main parent lineage of Delta, where we can see, do any of these have, or you can also search by Delta, which includes all the sublineages over here by itself. But we could also say, okay, for all the B1 617.2s, do any of them have the spike E484K mutation? So you can click on that, for example, wait for it to go. And there's 8. So out of the 18,000, you can get, so it's a good way to just get quick numbers. But you have to keep in mind, though, that this is data, only data that has been sequenced and only data that has been sequenced, passed, and then submitted. Sometimes it takes labs a bit of time to get data submitted. Some labs only submit once a week or once every 2 weeks. And so you might, for some of the newer data, you might not have a complete picture. So you always interpret with caution when it comes to looking at some of this data of maybe for the previous few weeks that not all the data is quite there and could change as people get things submitted. But that's a good way to quick search for Florida lineage, different mutations. If you have a particular accession, someone's giving you an accession and you need to pull down that sequence for some reason, you can search. Oh, you can also make sure you can click on the individual sample and you can get different information about this sample. So the virus name, accession, type, lineage. You also get the Scorpio call, which is part of the Penguin program. It'll give you all the different amino acid substitutions and just different information, the collection date. You can get this, how is this sequence, how is it analyzed, you can get the coverage, and more information about the originating lab, the submitting lab, and you get the sequence. You can either click here for the FASTA file, or let me copy this. Like pretend like I click on this and someone just handed me this accession. You can also, let me wait for this to reset. You could also paste that in here and search based on the accession. You can also search, this is the virus name, and we'll go over this more tomorrow about how you rename these and why it has to be renamed in this format, but you can search based on the virus name, host. So if you're looking for things, there's not only human samples in here, there's samples from like the cat. I think there was a tiger sequence that was sequenced from the Bronx too, that's on there. So there's different hosts. I think there's minks. You can search based on database on hosts. And so as you can see, this pulled up that accession. I'm gonna go back to just the Florida data. So you can also search based on collection date. So if you wanted to grab all the data from April of 2020, you could, search based on the dates. You could search if you're looking for particular samples that you submitted on a particular date. You can use these here. If you're trying to pull data for a particular study, do a particular analysis, but you want to make sure that you only have complete genomes with high coverage, you can click these different features here to filter out the data. So there's a number of different ways that you can do that. You can also click on this here. It'll check every single sample, so all 67,000. And let's say you wanted to download the metadata for those samples. So you wanted to get all the accessions, you wanted to get all the lineages, and what was going on for our state. You can click this download button. And you can click on sequences, so I can download all the FASTA files. You can click on, this is a bit misleading, but patient status metadata. This includes basically what's in this table with a little bit more information. It's the virus name, the accession, collection date. It also has the lineage, so that's a good way. That's what I use to click on if I'm trying to get lineage information. So it's not, because most people aren't submitting patient status information. Some do, but most are not. But you can also get information about the sequencing technology, so different things that you need. However, what I'm about to show you is I'm going to walk you through an example of how to pull out all the information from your state. So doing it this way, checking everything and then clicking download if you wanted to get all the lineages per se from your state. It only lets you download 10,000 at a time, and that's it. So if your state has more than 10,000 sequences in it, it will not let you do that. So for example, Florida has 67,000, I can't. I'm about to walk you through an example to show you how you can get that information and how you can use HiPerGator to then pull out your specific state information. I'm showing this to you. We get a request. We give our EPIs. I call them lineage polls. GISAID and NCBI both rerun the latest version of Penguin every single day on all the data that's in their databases. So because, as you know, Penguin updates frequently, lineages can change, some lineages can get discontinued. So if you ever want to get just kind of a snapshot, what's going on in your state, what's the most up-to-date information, we pull that information for our EPIs, and then they track back the virus names in our accessions back to the patient information that they have so that they can get lineage updates and whatnot. And I have all this instructions. I'm going to pull up the slides really quick. What I'm about to walk through, I have it step by step in this information across these next 3 slides of what to do, but I'm going to show you a demo of how to do it. So I call them, we do these weekly. I call them lineage polls. What you want to do is go under the download side. So this is only if your state has more than 10,000 sequences. Otherwise, you can just pull it by clicking here and doing this download button and clicking on the patient status metadata. That'll give you the same information I'm about to give you. But if you have more than 10,000 sequences, do this. So you go to the downloads button and then scroll down to the second section here where it says download packages and click on the metadata. Click that you agree to the terms of use. Download. And it might take a second. It's a large file. And what this file has is it has like 1 line per sample that is in GISAID. So it's going to be a file with over 3.5 million lines in it. And this has the sample name, the accession, it has the lineage, any other metadata associated with that sample is what's in this file. It's a large file, so it's a compressed file. And I'm going to show you how to drag it over to HiPerGator, or if you have another Linux workstation, working the command line to pull out the information in it. So it's been downloaded. So we can transfer it over to HiPerGator. So I open up WinSCP, log into HiPerGator. And I was having some network connection issues on my side earlier, so I'll see if the file transfers. If it doesn't, I've already transferred an older version, so I can show it to you. This doesn't work. So on the right side, I have my HiPerGator folders. On the left side, I have my local computer folders. So I've just made a space. You automatically log into your home, so you need to navigate to the blue folder or the blue drive. Go to your state folder, and then my username. And then I have a place, you can either make a folder here, wherever you're going to put it, and I have that in the instructions on the slides, or I put all of my SARS-CoV-2 information in a folder called data, SARS-CoV-2. Let's scroll down, and this is just kind of my space for doing this, so I can keep everything organized, all my GISAID lineage polls. So I already have an older file here. Let me see if I can get this to transfer. So you can either click down to go where you're going. I need to get into my downloads folders, which for some reason I can only get through this way, but otherwise you can click to it. And let's see if this transfers. All you do is you just drag and drop. If it doesn't transfer, I'll use the old version I have on there. I was some kind of network issue on my side. That was happening earlier today. It looks like it's happening again. So I will go ahead, pretend like it, yeah, I'm having a network issue. So pretend like it moved over, as it should. And I will just pull up PuTTY now. and just use the old, I'll just use the old file I have on there to do the example. You don't need to do this, I'm just doing this so you can see my font better. Let's share screen. So to log into HiPerGator, hpg.rc.ufl.edu, your username, password. It's easier for you to see. Okay. So now we need to navigate to that folder. So I have it in this folder here where I keep it. And I'll show you how I'm going to do, or yeah, I was trying to transfer that. Let me go ahead and remove that. Okay, there we go. So I'm going to show you how I'm going to remove this one as well. Just because. There we go. So these are some older versions. So when you transfer a file, this is the file that I transferred. I transferred yesterday. It's the same file that I just tried to transfer now. And it's going to be saved as metadata underscore TSV. And then it has the date that you pulled it from. And then it's got tar. Tar is just a way to compress and package files together. XZ is the type of compression. So first we need to uncompress the file. And again, I have this step-by-step in the slides for you. So there's a bash program called onXZ. And it's going to decompress the file. Take a second. Big file. Okay. So now if we list, now you'll see that it's uncompressed and now it's just a tar file. So now we need to untar the file. We do this like that. And that's going to basically unpackage it, and we'll be able to see the individual files that were within that. So you can see here we have our metadata file that we're interested in. It keeps the tar package, which we no longer need, and it has this README file, which basically just has GISAID's terms of use in it. We don't need that. So the file that we need is our metadata file. So I'm going to show you what this looks like. It's just TSV stands for tab separated, and it's just like a tab delimited file essentially. So it's hard to read like this. This is technically something you could open up in Excel, but it has this header line here. Oh, sorry. It has this header line here where it has virus name, type, accession for each one, and then you can see all the different values down below. So you can see it's starting out with some samples from Australia. For this purpose, for this example, I'm trying to show you how to pull out all the information related to your state. So I'm going to pull out all the Florida information from here. So I'm going to show you how to make a new file, pull out all the Florida information. We can also see in this, we can count the lines in this file. And we can see that they're in this particular one, because they usually make it, or this is the one from yesterday. So there's 3.4 million lines in this file, and so it's 1 line per sample. So first what we need to do is we want to make sure that we keep our header line, and then we want to make sure that we just pull out all, in this case, the Florida rule. So the first will be head -1, which is saying I want the first line in this file. So if you were to just do that and print it out, you see that it prints out just the header line from that file. So we can go ahead and stream that output, which is this greater than sign, into a new file that we want to save. So I recommend just naming it based on like today's date. And then I do like, I'll show you an example of how I name things like this. I just do my state lineages all just so I know what's in that file. Like that. So now I can see that I have that new file here, and if I were to print out the output from that file, I can see right now it just has the header line. So now I need to add in retrieve the Florida information from the big file. So we can do that using a command called grep, which looks for, it recognizes patterns, and what it's going to do, it's going to return each line that contains the pattern we're looking for. So we know that in the location field, it'll look just like this. Remember your spaces on either side. And then you type in the state you're looking for. In this case, we're looking for Florida. And then you want to do the file you're pulling that from. I'm going to, just to show you as an example, I'm going to pipe this. That's a pipe. It's going to stream that information into the next command, rest, and it just opens it up. So you can see right now I'm pulling in every single line. In this case, it has the location that sample came from Florida. So it's pulling in all the Florida information. So what I can do is then append that. So if you do 2 greater than signs, that appends to a file. If you were to just do the 1, it would overwrite the file we already have, and then you'd be missing your header. So we want to keep the header in our new file and then add in that Florida information. So that's our new file. We're going to append that information. Let it go. So now if we just peek into that file, we can see that we have our header, and then we can see that we have every single line starts with Florida. We can even now do a line count of that new file like this, and we can see that there's 63,000 over 63,148. So now we have a file with a header, it's tab separated, and has all the information for state of report. And you can do that for any kind of data that you're looking for. And all you have to do now is use WinSCP. It's going to let me log in. I probably won't be able to transfer it back. Try to transfer that back to your local computer. And then you can open it up in Excel. You can share it with your updates. You can share it with anyone. And I will open up a copy so you can see what it looks like. All you do is you just navigate back to that folder. There you go. And you'll see this file here, and you'll just click and you drag it over. Mine might not work right now because of some network issue. If you are still logged into WinSCP, you might not see the file here. You just need to make sure you click this refresh button, this green button, because if you're logged in and you make changes, it's not going to refresh automatically, so you need to do that. And then you just click and drag to whichever folder on your side that you want it to go, and then you have that. I'm going to open up this really quick. I'm going to open up Excel, and I'm going to open up I'm going to open up the old version that I've already made. Open an older sample from previous times, and I'm really quick. So it's opening up one I have here. It's just tab separated, so you can hit finish. Okay. So this is what it looks like. So this is a great way to be able to share information, what's going on in your state, getting the most up-to-date lineages. So you can see that you have virus names, this is everything from Florida. You can see location. This is a way to retrieve accessions, and then you can see over here that it has the GISAID code, but then it has the Penguin lineage, and then it also has the Penguin version. And so these get updated daily. So this is a great way to keep an up-to-date list and whatnot. And from here you can make figures, make graphs, charts, whatever you want, different counts, frequencies, based on what's in there. It also has additional information about amino acid substitution, if it's a complete genome or has high coverage, GC content and whatnot. So we do a lot of different stuff with this file. We pull it weekly in this day. So that's why as part of this training, I just wanted to show you how to do that, so that in case you ever wanted to retrieve that type of information about your state, that you're able to do that. So we'll go back to our slides. So as I mentioned, I have those step-by-step instructions on how to do that, and the reason why. So you have more than 10,000 genomes on both of these slides. So it's just step-by-step of exactly what to click on, how to transfer it, and then exactly each one of those batch commands that I did, change into your directory, list it, how to uncompress that file, how to untar it, how to retrieve that header line, and then how to append your state-specific information to that. We'll go back into presenter mode. And then last slide. So we're done for the training for today. The next training, so tomorrow, we'll be going over how to, on HiPerGator, it'll be a lot of live demos on how to run Vader on HiPerGator so that you can do sample review, and batch your samples based on if they passed or required in the review, and then how to generate your multi-FASTA file that's formatted and all of your samples renamed to their public names tomorrow. And then Friday, we'll go over, you'll have your FASTA files already made, and then we'll go over how to all the different metadata templates for GISAID and NCBI and walk you through step by step each one of their portals on how to submit that data. And then after that, if your jurisdiction would like, we can have a follow-up call with just your lab to go hands-on, walk through, share screens on your side with your own data. So if you have any questions, you're trying to troubleshoot or anything, we can get that taken care of, because I want to make sure that all states and jurisdictions in our region are able to, at a minimum, submit the high priority information, which is those consensus assemblies to GISAID and to NCBI, along with that sample metadata and what's required. And then after we get everyone is comfortable with submitting consensus assembly, we'll then go into the next trainings on how to submit those StaPH-B files and then go into the flag sample review, variant confirmation, and assembly correction, which Part 5 actually might, depending on how much time it takes up, it's quite a bit of information in there that might actually get split into a couple different trainings. As I mentioned at the beginning of the call, the recording for each one of these trainings, the slide, and any associated training materials. So for the next couple trainings, I'm going to be sharing some Excel templates that we use and other information. Those will all be available at that link there on our Southeast region GitHub site. So with that, if there are any questions, we have a little bit of time for questions. And also, if you think of something after the fact, please call us, send emails, anything like that. But I'll take questions now. And also feel free to put them in the chat. If you don't have a microphone or anything, feel free to write. There's a chat that you can write questions in. Okay. Well, if there's no questions, if you think of something else, feel free to email me. Otherwise, we'll go ahead and end this call. I'll stop the recording, and I'll talk to everybody tomorrow.